To introduce Dr. Jessica Metcalf today. She's an associate professor in the Ecology, Evolution and Public Affairs at Princeton University. She's been there since 2014. Her whole road started in Imperial College in London, where she got a PhD in 2005, working on the evolution of demography of, I just call it certain plants, because I didn't know what it was. Some kind of cool plants. And then she went on to do a whole bunch of different postdocs. She went to Max Planck Institute in Germany, where she studied senescence. And then she went to Duke, where she studied tree demographics. And then she went to Penn State, where we actually met, where she studied infectious diseases. So she's done, a lots of things under horizon, no one-trick pony whatsoever. And then she went on to Oxford, where she was a research fellow for a couple of years until Princeton scooped her up. Very clever move of them. So she's a demographer, and as I said, she has lots of different interests. This is just a small proportion of it, really, that she does. But Jess is absolutely wonderful person. She's, I would say, one of the cleverest people I've worked with and had the pleasure of working with. I just had a bunch of data, and she somehow made it into a science paper. It's like, I couldn't have done that. And then yeah, she had a whole bunch of science papers afterwards. So very productive scientist. I'm very glad that she wanted to come here and talk to us about this. So floor is yours.
Well, thanks, Sylvie, and thanks for having me. It's wonderful being here. My brother very carefully learned to say, my sister did a PhD on the evolutionary demography of monocarpic perennials. What does your sister do? As a sort of chat-up line, but I don't think it ever worked. So I don't think anyone who's in the same department as Melissa probably needs an introduction to the fact that there are 6 differences in immune function. I'm really going to be focusing on sex here, so chromosomal and hormonal differences rather than gender. But asking, sort of probing it, thinking about how these immune differences, what they look like and how they might have originated. And of course, the absolutely first thing you learn when you walk into this field is that we know strikingly little about sex differences in immunity. So in a lovely review, Sabra Klein describes how in 1977, the Food and Drug Administration decreed that you shouldn't do a clinical trial on a woman of childbearing potential. That's sort of everybody, pretty much. And I think in 1993, they decided that women might be human. But even in 2005, 8 out of 10 drugs that were withdrawn from the market were withdrawn because of side effects in women. So the first thing you learn is that we don't know anything, quite apart from the strange cultural maelstrom you're walking into when you start thinking about these questions. So I'm going to talk a little bit about, give you an example, a motivating example as to why these differences really matter from a point of view of sort of health. And then move from that to my true love, which is thinking about the evolutionary things that might have driven such differences, how they could have evolved. So start by thinking about the influenza virus. So this is a remarkably difficult challenge for our immune systems to work through, because it has all these surface proteins that keep changing all the time. And these will interact because of the way our immune system learns about them, and then they change. You end up with this very complicated life course of immunity to influenza. A lovely study by my colleagues, Justin Lessler and many others, from a cross-sectional survey in Guangzhou province in China, looked at all these different strains of flu. And so this is moving across different eras of different flu circulating strains. This is Hong Kong 68, Victoria 75. New flu strains emerge, your immune system learns about them, and then they kind of get replaced by the next strain. And when your immune system learns about a flu strain, you can detect sort of how it's learned in some sense by looking at the antibody titers in your blood. So how much if you were to expose the antigen associated with each of these different circulating strains of flu to an individual's blood, you would see how much the response would be, how big the response would be. And that's what's shown on this plot. For individuals of very different ages, you're seeing the darkness of the color reflects the antibody titer response. And so this study happened in 2014, anyone who was below the age of around 30 in 2014 was too young to have seen the 1968 circulating strain. And that's why you get this sort of triangle of no response at this pattern. there's this also really fascinating thing where if you look at the age you were at which a virus was isolated, so this differs as you age through life, you find that your immune system becomes obsessed with the flu strain that emerged when you were around 7 years old. Like whatever strains you show it afterwards, it's still reacting as if it saw that first one most passionately. So kind of fascinating. Many interesting sort of consequences for thinking about how we design vaccines, etc. But what I was interested in was like, this all seems very nice, but what happens when we also add sex into the mix? Because no one had really done that in the circumstance, but there was good reason to believe that this might result in different patterns. And if you do that, so that's showing the effect of the age at which the virus you were when that virus strain emerged. And this is showing you the sex differences, just fitting that all within the same generalized additive model. And so this is a virus, a negative number means that that viral strain emerged before you were born. And you can see that females, if it's below the line, that means that females have a larger response. So this suggests that females have greater cross-reactive antibodies. Our immune systems can recognize things that circulated that they had never seen before, or at least respond to them more than males do. And then you get this really wild pattern where there's really big differences also after menopause, which is fascinating, and for which there is, I think, it's not quite clear how that might have emerged. We expect, I mean, in general, you expect everything to fall apart at late ages, especially if you're an evolutionary biologist. So how do we interpret this particular pattern? And one of the issues, in going from these antibody titers to actual health is that it's not clear what the mapping should precisely be. Another layer of information that they had from this particular study was that they actually had a nested case control study of 100 sex and age-matched individuals. So we could think about, like, is it good or is it bad to have a high antibody titer? Not necessarily clear, given how intricate the immune system can be. So here I'm showing you again the year of circulation of the virus strain, and we just looked at the log odds increase in mortality for all these different strains. The first, and so if a point comes out above the line, that means that mortality went up with an increase in antibody titer. If a point is below the line, then mortality went down. And this is the pattern that you see. This study was not powered for this analysis, so nothing is, inverted commas, significant, but the magnitude of effect sizes is really shocking. So this suggests that there's a 20% increase in survival, reduction in mortality for a 1 titer increase in antibodies. Astonishing, right? And there's no way that can be quite correct, because 20% of individuals are not dying of flu at late ages, right? There are other causes of death out there. It might be that influenza antibody titers are a correlate of more general protection, but I also think it's really striking that we don't actually know this. So we've done this analysis right around the time where I was getting interested in sex differences. And so, of course, the next thing to do was to break it down by sex. And then it becomes even more weird and interesting, because you find that in general, in males, 1 increase in log antibody titer increases survival, so mortality is reduced. But in females, if anything, the opposite is happening. And you can come up with some reasons as to, maybe there's actually, again, it's hard to say whether nothing's happening because of the power of this analysis. It's possible that just because of nonlinear averaging, for females, you're higher to the sort of top of the line there, so there's no benefit from an increase in antibody titers. Females generally just survive better, whereas males are lower, and so an increase in antibody titers would increase male survival. It's possible that females are bringing many different weapons to this war. So we know that cellular mediated immunity might be stronger in females. So they might be, you might not be able to see that trajectory, that increase of survival with antibody titers in females, because so many other things are in play. Or it might be that it's frankly bad for females to have antibody titers, high antibody titers. And an important piece here is that the immune system is a really dangerous line of defense, right? Having a high antibody titer, having, all those, whenever you look at those probiotics that boost your immune system, I think, God, I don't want that, right? Like every autoimmune disease, every way in which so many of the manifestations of COVID were associated with your immune system being deregulated. An immune system is a dangerous line of defense, and it could be that a higher antibody titer is even higher in females, leads to even worse outcomes in females. So we care about these sorts of differences, obviously, because influenza is one of the sort of very large killers, but it's also sort of fascinating and puzzling to identify that you get this much larger, sort of broader cross-reactivity in females relative to males. So to unpack that a little bit, I'm going to switch gears and ask now, in a larger evolutionary context, why might we expect such sex differences in immunity to evolve? And if you're an evolutionary biologist, the conventional narrative is quite simple. males are expending resources on silly things like peacock tails or fighting or whatever it is. And thus males are the weaker sex. So this might channel resources away from immunity, and thus you might have a lower responsiveness to infectious diseases, etc. And to put some conceptual bones on this narrative, this perspective actually came from a very robustly empirical approach. It wasn't just theory. In 1948, inspired by Darwin's views on sexual selection, a man called Angus John Bateman published an experiment where he measured the fitness of flies across many generations and found that males' fitness increased with the number of matings they acquired, whereas females' fitness increased with the number of times they actually reproduced. So the thought then was quite simply that males should invest more in immunity. But it turns out that, of course, it's more subtle than this. And Hannah Coco and Michael Jenians unpacked this a little bit in a paper they wrote in the Journal of Evolutionary Biology. And they pointed out that more immunity doesn't necessarily translate into greater longevity. There's lots of other ways to die, right? You might be channeling resources away from another way of surviving. And longevity doesn't necessarily translate into higher fitness. So fitness is going to require both survival and reproduction. So this sort of simplification might be a little ineffective in probing these sex differences in immune function. There are a lot of other things going on. One of the challenges, what can we learn about immune system function? One of the challenges in this area, as in so many areas, is that most of our data is from species like this one. This is my friend Justin Lesser's baby, which is a fairly narrow lens, right? We're a pretty odd species in many different ways. And the question here is, I'd love to understand the differences in how sex differences might shape immune functioning across the tree of life. But most of what I know comes from this species here, which has a bunch of different characteristics. So we have determinate growth, we're senescent, we have to care for our young, we're primates who are big-brained and long-lived. But even within that, we're very weird primates. We live twice as long as you might expect. Maternal mortality seems to be incredibly high. Birth intervals are short, we have menopause. There's a lot of reasons why it might be tricky to grapple with thinking about the broad swathe of diversity by using the lens of humans. But nevertheless, keeping this in mind, I'm going to walk forwards based on what we know and try and probe what I might expect in terms of evolutionary outcomes for sex differences in immune function. And of course, there are two drivers, as I mentioned before. The first will be chromosomes. This is the karyotype of a human male. So the Y chromosome you can see there, whereas females would be XX. And one of the astonishing things you discover as you start learning about this area is that tons and tons of immune genes actually sit on the X chromosome. So many of the pattern recognition receptors, like the toll-like receptors, toll-like receptor 7 and 8 sit on the X chromosome, but also FOXP3, which tells T regulatory cells, so the sort of hemostat cells that tell the immune system to calm down, that also sits on the X chromosome. And some areas of the X chromosomes, as Melissa has shown, are not as efficiently silenced, and so you could end up with a double. So the way that dosage compensation works in humans is that one of the X's in females is just switched off, but the switching off happens at random, so you can end up with both greater diversity in females, but the switching off also isn't complete. So sometimes you can end up with 2 copies of a gene that would only be single in males. And toll-like receptor 7, one of these pattern recognition receptors that is crucial to innate immunity, sits in one of these regions and has been shown to both be critical for defense against influenza, but also drives autoimmunity. So it seems like that there's a lot going on in terms of how the immune system is organized around the sex chromosomes. Both good and bad effects, which rather foreshadows where I'm going next. And there's also some really fascinating stuff about how being heterogametic doesn't necessarily mean that you're male, right? So this is another interesting lens onto this problem. You can be heterogametic, and if you're a bird, it's the complete reverse of mammals. And if you're a random tetrapod, if you're somewhere in the frogs or salamanders, you can actually be a sort of mishmash between the two. And what you find if you do an analysis, this was by Pippolo et al. In Nature, is that it looks like survival up to maturity is just worse if you're heterogametic. Right? So there's probably other things going on, not just about immunity, not just about being male versus female in terms of how much you're contributing, how you're contributing to reproduction. So there might be a piece of the sex differences that is simply due to the fact that, being the heterogametic sex is not so good for you. Chromosomes, half the story. The other half of the story is, of course, hormones. And every single one of the immune cells that circulate around your body is completely in hormone receptors, it turns out. Estrogen receptors, progesterone receptors, androgen receptors, they're just, every single one is responding intricately to the hormones that are circulating. And if we think about how these two pieces, these two mechanisms, the chromosomes and hormones, might add together to translate into an immune function, there are two ways in which you can sort of crudely, you can build a cartoon of how the immune system works by breaking it down into two different pieces. So the first is detection of that a pathogen is there at all. And the second you can think of as response. Right? So the detection, all of the pieces of the adaptive immune system, all of the toll-like receptor stuff, so your kind of encoded pattern recognition receptors, the whole suite of mechanisms that are there to see if a pathogen is actually present will play into this first trade-off. And then the response might be just, how many macrophages you send to the front, how large is your response going to be? And so with these two differences in mind, I set about to read about the immune system, which is a very punitive thing to do. And took a long time. And fortunately, Sabra Klein has summarized this repeatedly. But what you find out is that there are very consistent and sort of interesting differences that kind of fall out alongside this organization. So females seem to have more CD4 T cells to CD8. They have more immunoglobulins. They have more efficient antibody-presenting cells. All things which seem to line up with detection and discrimination. So they have more, more little messengers that are traveling around carrying bits of potential pathogens in the mix. They have more B cells. If you look at the sort of T cell school that tells T cells how much they can tolerate, the school in females seems to be more lenient, so you get a broader response. Everything seems to suggest that females are much more on the I must detect scale. Again, this is my cartoon of how the immune system works. It's not necessarily reality. On the other hand, males have more natural killer cells. They have more CD8 plus cells. So these are the kind of killer T cells. They have higher toll-like reception 4 expression on macrophages. They have more neutrophils. So you can put this under, it looks like males are much more going for the response. So maybe they're not detecting pathogens so often, but when they do detect them, they start a huge response. There's more sepsis in males, for example, as well. That seems to happen more often. So I'm not saying this is the only way to interpret what's going on, but it's one that at least seems to be one way of interpreting this crazy complexity of what's going on. So how do we work with these two trade-offs? Let me give you an example of how discrimination might work. So if you imagine some variable like CpG content, this is C's followed by G's in the genome, and this is actually a really good tell for a pathogen. And that's because we have a relatively low CpG content for reasons to do with how methylation works, etc. So if your immune system just goes around looking for things which have a high CpG content, there's a very high chance that that thing will be non-self, will be some sort of pathogen, something's gone wrong. And of course, if these two distributions were nicely separated, it would be pretty straightforward. You could assume that everything that was below this line was you shouldn't respond, and everything that was above the line was a baddie, and you should respond. But of course, many pathogens evolve and evolve mimicry. They want to look as much like us as possible. Or, the discrimination might not be that good. And so when you drew the line in the world where there's an overlap between these two distributions, you end up with a very classic trade-off between false positives and false negatives. And in the context that we're thinking about, a false positive means that, you've drawn the line here. You said I react to everything above this line. Then sometimes when I react, I'm actually going to be reacting to self, right? And that results will translate into immunopathology. If you move further over the line further over to the left, then you have no danger of immunopathology. You're not reacting to yourself, but you do have some false negatives. And there you might be at risk of disease mortality. So this is a trade-off I learned about because they made me teach epidemiology. And in epidemiology, they refer to this as the trade-off between sensitivity and specificity. And they usually draw the curve this way around, I'm not quite sure, purely to confuse people, sensitivity against 1 minus specificity. And the way this shakes out is sensitivity is the probability of having true positives. So there's a chance you respond if the thing actually is a pathogen. So for example, if you were to draw the line over here, you're not responding to a whole bunch of the pathogen area, the red bars, and so your sensitivity is low, but your specificity is high because you are catching you are never reacting to yourself. And as you move across, you move your response threshold across this curve, you end up moving across the spectrum from false positives to false negatives. Okay, false negatives to false positives. And this, if you're an evolutionary biologist, is fantastic, because then we have a question we can ask, which is, where should you draw the line? You always need a trade-off to ask these sorts of questions, because otherwise the answer is just survive as much as possible, have as many babies as possible. And so regrettably, the way this shakes out in the language of epidemiologists is that males seem to have, based on the way I have read the immune literature, increased specificity. So they don't react to self that much, whereas females are the sensitive sex. I don't love that. There is increased sensitivity, i.e. that you tend to always respond appropriately to pathogens, but you sometimes also respond inappropriately to self. And this lines up also with the fact that 80% of autoimmunity is concentrated in women. Surprising. Very surprising if you're an evolutionary biologist, right? Because you'd think that if you were essentially carrying a pathogen to term, you would have less likelihood of responding to it. So that's the first trade-off. And the second trade-off is in terms of magnitude of response. And this one, I think, is pretty easy to understand. You can imagine that if you don't respond at all, then you might have a really big chance of dying due to the infection, but you would have very little immunopathology. Whereas if you responded massively, then you would have vast immunopathology, but not that much infection hazard. So I can take my two trade-offs. And again, my expectation there is that males should be higher in the response level based on these various tells from reading about the immune system in males. So you can take these two relationships and put them together. So that what we're expecting is that as sensitivity goes up, there's an increase in survival, but then a decline, right? If you're responding too much to everything, then you're actually hurting yourself through immunopathology. If you're responding too little, you're hurting yourself through infectious diseases. And somewhere in the middle, there's an optimal. And likewise, as infection hazard goes up, you expect to have some sort of intermediate optimal. You can write this down in a pretty simple analytical model and build a sort of fitness landscape where on the x-axis, I'm showing you that magnitude of response. And on the y-axis, I'm showing you the discrimination trade-off going from false negatives, so not sensitive, to false positives, super sensitive. And I will show you on this surface an estimate of fitness taken as an estimate taken via their effects on survival. And this pattern is super robust to pretty much whatever parameters you put into it, which you find is not super surprising. It's that if you are something that goes for the really massive response, you tend to be something that doesn't respond all the time. Whereas if you're something that goes for a really tiny response, you tend to respond much more often. So there's a sort of balancing out in terms of how you react. So at first I was like, this is great. I have two sexes. I have two different optima. My work here is done. And then I realized that based on this picture, there's actually no reason for females to be the more sensitive sex, right? There are two optima, but there's no discrimination between which sex should end up on which peak. So I went back to the drawing board and I started thinking about, is it possible that males having less resources to expend on immunity could explain the fact that males end up being the less sensitive sex? And you could put that into the model relatively simply by assuming that males had sloppier discrimination between the two, so they weren't as good as seeing the difference between the two distributions, or that they made smaller investments, or that the investments were more costly for them. The trouble was that with both these, putting both these pieces in doesn't actually give you the answer that you want. It ended up making males the more sensitive sex. So there's something wrong with this framing, obviously. One obvious piece that might have occurred to you is that there is no, it is a super simple model. There's not a lot in there about pregnancy, etc. But before going that far, it's also a very simple model in terms of really making assumptions about just kind of thinking about it in the context of innate immunity. But there was one more piece that I thought, in thinking around how a deviation away from innate immunity might result in more funky results. And that was the role of, first of all, thinking about how immunity plays out across the life course. And of course, being male and female is not just about surviving, it's also about reproduction. And there are different investments made across the life course, and these two sexes are subject to very different risks. And so here you can take this basic framework and you can insert it into a more classic life history evolution framework. My favorite definition of life history evolution is it's an elaborate answer to the simple question of why having more offspring is not always selected for. And what you require to answer that question is a model of survival and reproduction, a model that can give you fitness based on age trajectories of survival and age trajectories of fertility. So an estimate of fitness might be something like R0, the net reproductive rate, and that can be calculated by taking survival up to every age, so this is survivorship, times the number of offspring produced at that age and summing that across the life course. And so then we can pick apart like what's going on that's different in these two circumstances. So females get pregnant. Pregnancy might be associated with higher mortality. It might be associated with higher infection. It might be associated with higher infection-associated mortality. Only the latter gives you the increased selection for sensitivity that I predicted. If you put in immunity effects during pregnancy, so my colleague Andrea Graham has done some lovely work showing that Soay sheep on the island of Soay, the island of Soay, the females that are more prone to miscarriages also survive more, and they tend to show markers of an autoimmune antibody. So it seems like having a really powerful immune system, I mean, this really speaks to that, this being how the functioning of the immune system works, right? And females do seem to be more sensitive. They're reacting more to self, but the cost of reacting more, perhaps generally, is that if you're reacting more to non-self, you can end up with more miscarriages, but you might be better protected against pathogens. This does not particularly explain or set you up in a circumstance where you can end up with females on the more sensitive peak. There are other little nuances, like if you start thinking about the fact that many vertebrates have the way that the benefits accrue over the life course might be different for males and females. So in particular species where males accrue harems and their real reproductive success plays out much later in life, then you actually do get selection towards lower sensitivity. And that's because every year you're alive, you're paying a cost of false positives. So if that late life is much more important, then you might actually have lower sensitivity to make sure you get there. But none of these are terribly satisfying as explanations for what's going on. They're all sort of a little bit bitty and they only work in certain circumstances. And so I was sort of thinking, is there a way in which by not thinking much about adaptive immunity, we're missing a big part of this story? And one of the really important ways in which adaptive immunity might be super different between males and females is in thinking about maternal immunity. So adaptive immunity means that you can generate memory to pathogens you've seen, and then you can respond efficiently to them in the future. But a core difference between the sexes in this regard is that females pass that information on to their offspring. So anyone in this room has almost certainly either had measles or had the measles vaccination. And if you're a female that produces a baby, that baby will be protected for its first year of life from antibodies from the mother. So they're passively acquired. They just pass through the placenta or through milk, and they keep the child protected from infection with measles in that first year of life. And this is hugely important for survival in that very vulnerable first part of life. So it's probably really important for fitness. Building a model that includes adaptive immunity is, so I'd sort of built pretty simple analytical models at this point. It's actually really, really hard to do that when you are thinking about adaptive immunity, because suddenly you have to remember things, you have to remember a diversity of things. So what I did to grapple with this was I built a relatively simple agent-based model where I had different individuals, and I basically threw a stochastic set of potential pathogens at these individuals. And then they responded to the ones, they protected against the ones that were above the line. They had immunopathology for ones that, in certain circumstances based on what they were, and circled around it. And just as a sanity check, I made sure that as you looked across this, again, we're talking about where you draw the line, looked across a spectrum of that threshold in terms of survival within the stochastic model, what you got, and what you get is that intermediate fitness landscape I described before. The different line colors show increasing disease hazard. So if the disease is really, really bad, then drawing the line very far to the right and thus ignoring all of the pathogen space is really bad and you die a lot. Less so if the disease is less bad. And then this spectrum shows the different magnitudes of immunopathology. So the basic conceptual outcome is the same. You get an optimum that is defined by the hazards or the risks of dying accrued through immunopathology or through infectious diseases. And now we can take this basic framework and say, all right, what happens if we add in offspring? So going again, I'm going to assume the way I'm going to work this is I'm going to assume that kids acquire the threshold of their mother. And the first assumption to make is that the threshold, the children get nothing from their mother. If children get nothing from their mother, then children have no benefit based on where their mother draws the line. But if what children get from their mother, so as the disease hazard goes up, children survive less. But if what they get is protection from their mother without paying the cost of immunopathology, because presumably, these are just circulating antibodies that are passively there in the blood. There's not a lot of, machinery that's destroying your own cells, etc. Then you end up with selection towards greater sensitivity because you end up with this benefit, this sort of considerable benefit of having a higher range of reactivity. So this would argue that maternal immunity seems to be an important part of how you might end up with greater sensitivity in females relative to males. And we do see, there is an interesting question, which is how do you actually test this or probe this issue? So pieces of it are certainly evident. So it is certainly the case that if you look at vaccination by homologous or heterologous vaccines to protect offspring from infection, you do get sort of differential and interestingly differential patterns across in flu, at least, and across different species. But I think the real exciting axis to test this would be to look at one of my new favorite species, which is the cygnathids. So these are the pipefish. And the pipefish, very excitingly, have a whole spectrum of life histories that go from females just tossing their eggs onto the ground, or females gluing the eggs to males' tails, or males having a kind of little pockets that the females put the eggs into, through to males having a full, basically gas exchange, antibody transfer, like a pouch that is essentially the only other time that pregnancy, or the only other time that pregnancy has evolved within males. So there is an astonishing potential here to start probing whether these patterns that are suggesting, and it has also been shown, just to sort of fill this in, if you expose the males to a bacteria like Vibrio cholera, then their kids are protected. So there is transfer of antibodies through the placenta equivalents within these fish. So I think it's a super exciting case for me to write grants, still unfunded grants. But in as much as the work that has already gone, wonderful colleague Olivia Roth has started driving this work forwards, and it does look like male seahorses have a more female-like immune system than one would expect. So I think there is the potential there. The issue is, of course, the cartoons I've built require a certain design in terms of how you measure and what you measure. So it's a very fascinating set of questions, but just sort of, and I do think that the more we understand why these things might have evolved, and I'm not saying that this is the only answer, this is one potential, and potentially many things could be core explanatory variables, but I think the more we understand them, the better we are equipped to walk forwards into thinking about how these, how the sort of public health and medicine outcomes of these pieces might play a role. And so just to give you a couple, I'll give you a couple of examples of this from some of the more virusy end of my work. My favorite definition of a virus is from Medawar, which is a piece of bad news wrapped in a protein. And in the influenza virus, as you know, is very bad news indeed. There are 36 million symptomatic illnesses in a year and perhaps 90,000 deaths. But the news is really variably bad across the life course. And the reason it's variably bad across the life course, as I hinted earlier, is partly because of viral evolution. New viral strains emerge all the time. It's partly because of immune dynamics, our immune system learning and forgetting, but also our immune system senescing. And it's also partly because of population level effects on risk. So what's happening at this is always true for infectious diseases, what happens at the scale of the population defines the risk that I'm at. And so we did some work again on a more recent iteration of this Guangzhou cross-sectional study looking at antibody titers to different flu strains ordered by year of circulation. So again, there's this, the response is very low for individuals who were born after the virus circulated, so that's shown in blue, and titers, so antibody titers are higher for individuals who circulated afterwards. But if you lay out the most recent iteration of the data, you do see this, the lower quadrant of blue, but also you might be able to see there's a sort of suggestion of waves and oscillations through this. And we have a sense of what the mechanisms underpinning these oscillations. So it's sort of like, there's these waves of viruses are flowing through the population, and they're shaping waves of antibody titers in the population as well, right? Increases and declines. And probably the way it happens is you have your first influenza strain infection. There's an antibody response, it then wanes. There's another response. There's maybe what we refer to as back boosting. So some titers that look, some antibodies that look a little bit like the new strain are also boosted by the new strain. You get waning again. It might be a failed infection because titers were so high. And then there might be cross-reactivity that comes into play. And as I suggested, this might matter because cross-reactivity might be very different in females. If you put together all these different pieces, so boosting, this original antigenic sin symbol, waning, and infection risk, you end up with a simulation, a really simple simulation that actually echoes these long-term 20-year cycles that we were starting to see in those antibody titers. So it's very cool, but there's obviously something intriguing and potentially important missing, which is the role that sex differences might play in shaping this. So the reason that we really care about this is because antibody titers are the one thing that we really can change, and we can change them by vaccination. There's reason to expect that where you sit, if you're very high, there's no point in vaccinating someone who's at a very high titer. To a first approximation, right? Everything's going to be somewhat context specific, but there is a way in which that might not contribute to their immune response. And there also might be differences in how your health outcomes were shifted by your antibody titer based on what sex you were. So because of both this differential response to vaccination, this differential life trajectory of titers embedded in differences in the immune system, our capacity to move them around, and the existence of differential outcomes in terms of whether you go to hospital or not, I think there's an awful lot of motivation for unpacking how these sex differences work and what they actually look like. That's for influenza. Influenza is certainly an exciting killer. Perhaps more exciting or familiar piece of bad news was the new bad news that occurred when the SARS-CoV-2 pathogen emerged within human populations. And in this circumstance, I'm sure all of us are familiar with the fact that there was a very striking increase among people who were infected. This is showing age on the x-axis and the number of hospitalizations on the y-axis. And perhaps the most immediately striking pattern was an increase in mortality or hospitalizations, ICU, and deaths with age, right? So mortality is concentrated in late ages. There's also a difference across sex. And we were interested in the ways in which these two pieces manifested across age and sex. We were also interested in just thinking very generally about how mortality of infectious diseases or other causes played out across the life course, both for endemic conditions, but also for these sorts of new emergent pathogens within the population. So I worked with Santé Publique France to look at data that they had on sort of all causes of mortality on influenza, and also on the emergent coronaviruses. And what you get, this is something that is incredibly familiar to demographers, you end up with a bathtub shape of mortality. I should say that I've also plotted these all on a relative scale, so they're all standardized to mortality at age 60. And in general, mortality is high if you're very young, then it comes down, and then there's a kind of flat bathtub area, and then it goes up at late ages as a result of senescence. This is a very, very familiar pattern. The range of the y-axis that I've plotted might suggest to you what's going to happen next. And that is that if you add SARS-2, or indeed this Middle Eastern respiratory syndrome, this is a different coronavirus, but also one that is emergent and worrying, what you find is that you end up with this sort of that bathtub bottom is totally lost. So you get this very high mortality of the young, it then declines until around puberty, and then it starts coming up. And if you're a evolutionary biologist, you might think, well, that is sort of, one might expect the evolution of senescence to be rooted around the early ages of reproduction, but we're not seeing it in the other species. So I think there's an awful lot more to unpack there. There definitely does seem to be a suggestion that things like influenza that circulate all the time, this bathtub shape is born from the fact that adaptive immunity is kind of flattening it out. Is after you've lived through your first couple of infections, you're saved from mortality by your adaptive immune system. I think it's really interesting the ways in which adaptive immunity works differently from other causes of mortality, and thinking about how the senescence evolves. And then it kind of increases again at late ages as a result of senescence. These new things, you have none of that shielding. Adaptive immunity is not saving you, so you end up with this very, very distinctive pattern. So that's for age, but obviously I'm interested also in sex. And so if you take the same data, but now you look at the pattern of mortality in terms of males and females across age. So what I'm going to show you is age on the x-axis, and then the relative risk of hospitalization. So if it's above the line, it's higher in males. If it's below the line, it's higher in females, for all these different infections in the same sort of way. And if you do that, what you get is a trajectory that looks like this. Males die more in general, right? So they're generally above the line, but during between the ages of 15 and 60, you get more infections of things like SARS-2 and things like influenza. Okay? People often refer to this as the childbearing years, but not all of us wanted to bear children, so so I don't. But I think this is so fascinating. I mean, I think we wrote a very descriptive paper about this, but why you end up with this flip-flop between higher risks during those ages and in females, and then the declines in males is really fascinating, right? Is it that males are senescing faster, so that's why mortality is going up so fast in males? That's really a question that one of the answers that has been suggested by some of the analysis that people have done. Or is it that female senescing actually blunts inappropriate mortality? We know that females tend to suffer from these immunopathologies, from these too larger responses to infection. So maybe senescence is actually, a little bit of senescence is a good thing if you're a female and you have an overexcited immune system. So I think this is such an interesting area, and I think a very productive area for sort of probing into how these pieces work out. Evolutionary history certainly shapes the way our immune system functions, and this will shape how individual outcomes come out. And then you end up with these population trajectories of infectious disease, which are the manifestation of the individual outcomes that will in turn shape the selection pressures on our immune system. But it's a very hard problem. So I think the way to sort of tackle it, and the way we've been tackling it, is by thinking about whether you can use fundamental principles from evolutionary biology to kind of shape out what you might expect selection pressures to yield, and semi-mechanistic models of what might be going on, probing existing patterns based on what we've seen in patterns of mortality across age and across time. Yeah, very exciting. So these are some of the papers I listed. That one's the basic theory in a review. This is on the differences in flu mortality across antibody titers. This is the cycling, and then that's the last bit with Santé Publique France. So I'd like to thank all my co-authors, and thank you for listening.
Thank you very much. Any questions? Thank you. That was a really nice talk. Going back, I have a question on one of the graphs you were showing of the titers for influenza and the suggested waves. Yes. And you suggested some mechanisms for why there might be some waves of increase over time. I was wondering if you've looked into, or others have looked into, the severity of those specific strains, and if that correlated at all to the response.
There's a real, one thing, of course, you never reveal all the truth when you but one of the real challenges here is you have to, so this is not about the severity expression, but you have to normalize by the underlying responsiveness of the titan. So it's a lab-based assay problem. So the height of these waves I couldn't speak to. The mortality piece, I mean, I think when a new strain emerges, there's generally a lot of mortality, there's not a lot of immunity. I don't know that we know a vast amount about the, you always have this problem with infectious diseases where we never know what the denominator should be because people who are asymptomatic don't turn up to hospital. So I don't actually have a good sense of what the relative mortality of these different strains are. I'm guessing that 2009 was a real problem. 14, 9, which is a low year. So I'm not sure, but that's a great idea. I imagine you're thinking about selection pressures and selective loss and how that would play out in this.
Yeah, exactly. Thank you. Hi. I had a question about where the figure where you were showing SARS and MERS, and I was just wondering that way. They're quite different, right, in terms of how they emerge, and I guess I don't really know about MERS and how something like if there's an epidemic or the pandemic versus something that has become like influenza, kind of chronic, how you would expect that would shape the shape of these lines.
So actually, I think that was the core of the question. So Middle Eastern respiratory syndrome there's been something like maybe 5,000 human cases ever. And the way it happens is that it comes out of camels. It seems to be a really great infection of camels. And it's not a great infection, well, from its perspective, it's not a great infection of humans because it's so deadly. People die really quickly. So there's maybe some tiny stuttering chains of transmission where a few people in a household are infected. So I think there is absolutely, your intuition is correct. There is extraordinarily unlikely to be any immunity within human populations to MERS. Now, there is a really interesting question. I sort of hinted at cross-reactivity being critical in flu. It's very important in many infectious diseases. And there is a question about, one of my very clever postdocs realized, why are there only 4 endemic coronaviruses? We know that there's been at least 3 spillovers in the last 20 years. And so if that pace had continued for 100 years, we should have 15 at least, but we only seem to have 4. So, and you can think through various reasons why that might be. Maybe we just haven't handled enough, that seems unlikely, maybe spillover is increasing in rates, that's possible too. But it does seem very likely that one of the things that's keeping new coronaviruses out is an immune barrier from the endemic coronaviruses. So it's possible that the endemic coronaviruses are doing, so these are the common cold infections, right, are doing something to the emergent ones, but you certainly can't see it here, right? Like an endemic coronavirus will tend to have, any endemic infection, you tend to, like, as children get older, they develop immunity. So you end up having this sort of very like high risk and then this flattening out in a single second group. So, and I think the strangest thing about SARS-CoV-2, it was so frightening at the time. I think the strangest thing about it was that we were all susceptible to it. So it really, that flattening out that you get, thanks to immunity developed from when you were younger, was just not there. I think there's really interesting questions about the fact that, like for children, every infection is an emergent infection. So the child immune system is presumably designed to both respond and be kind of cool about it. Because the way that SARS-2 killed people was very much associated with immunopathology. You were dead, you were dying because of things that happened after the virus had appeared. And presumably part of what happens in early life is the childhood immune system knows how to grapple with that, but also designs memory so it doesn't have to happen later. Very long answer to a first question, sorry. Not great, thank you.
Thanks. So, the females being the more sensitive immune responsive sex, right, is in part due to the fact that they pass those antibodies off to their offspring during these early years where they're really, really important. Are there species where that doesn't happen, and how does that? I know you're sort of getting at that with the pipefish, right? But is there evidence of that in some other species where they don't pass on those antibodies, and if so, would females then be less sensitive immunologically?
So the things we know about things like even birds do pass on some antibodies right through eggs and eggshells and stuff. So I think that's a great point, and I wish we knew more about sex differences and all these different processes. But it's hard to anchor at the moment in any. That can't be the whole story, because things like Tribolian beetles do have sex differences in immune function. But I don't know enough about tribonian beetles to tell you whether this lines up with sensitivity versus specificity, right? Like, there just isn't quite the matching between the two. And they also actually know I'm doing that because there's this fantastic experiment. The males wound the females during mating. There's a sexual selection piece, so the females have to ramp up all of their immune responses. So it's quite specific example thing. And if you remove that competition and that, then you can do experimental evolution away from that sex difference. So I guess what I'm saying is there are also many potential causes for sex differences. This is not necessarily the only piece.
Is the population you built these plots off of, are they industrialized populations?
This really was just santé publique française, all of the French health system work. So
Do you know if the shape is different when you look at different global populations?
actually, and I should take that back. This was in Saudi Arabia and other places like that. So these pieces were from France. This was from every data. I think this was actually also based in France, but the SARS pattern did look pretty much like that in every human population they looked at it. But I think the Middle Eastern respiratory syndrome is pretty restricted.
And stratify women into those that have had pregnancies based on parity. So if you stratify by parity, do you see any difference?
So we did look at that in the French populations. That is such a minute. I don't think we focused on identifying the degree to which whether they were detectable. Did we test for this? I'm not actually sure. I should go back and check. We certainly didn't. Perhaps you may have forgotten. But our there weren't enough people who had been pregnant and pushed this around the infection.
I guess I would expect that in natural fertility populations, you wouldn't expect to see the sex difference because we don't see that in immune function as much. There's much smaller, and in wild populations, the sex difference in immune function is there, but it's not nearly as exacerbated as in industrialized human populations.
I think it's really, I think there's so much to be done by like putting together parity and other characteristics with these outcomes.
Thank you. I was wondering for the framework with passing adaptive immunity to the fetus. And I was trying to think, okay, if we add immunosenescence with the age of cessation with menopause, and I have no idea, maybe you thought already, like, what do you expect? Do you expect this sensitivity to decay especially fast in women because there is no selection if we're talking very long term? But at the same time, they have more immune-related autoimmune diseases in their life. So did you already try to unpack those two mechanisms?
I haven't really thought, I'd love to have, I feel like, it's certainly a space where it would be wonderful to have more data also. There is I think that I'm quite convinced that menopause evolved because childbirth is so dangerous. And because children don't survive if their mother isn't around, right? So it makes sense to stop reproducing and raise those last 15 children you had. So if you're unlucky, I don't think that menopause is like a switching off of selection on females particularly. I do think, I showed right at the beginning that pattern where something really different happens around menopause, and I also told you that every immune cell is covered in hormone receptors. So I would really expect something wild and interesting to happen both mechanistically and evolutionarily. I don't think there's much done on it at all, right? And I would be, from the theoretical side, I think that the immune system is such a complex system of interacting parts. And senescence, we're talking about how things fall apart. So if you don't know quite how they work, figuring out how they fall apart is kind of intricate. I think many lines do suggest that we talk a lot about this. Actually, Noah and I were at a workshop together. There's some sort of fascinating questions about whether just being female means that you're better at homeostasis because you're going through these fluctuations and massive things like. And so perhaps like losing control of your immune system. And there's this very famous, actually, you probably know about this, but there's this late life health mortality paradox where at late ages, by every measure, females are in worse health, right? So they stand up more slowly, their handshake strength is weaker, they self-report as feeling terrible. Whereas males are saying, I feel fine, and they die. So how did that happen, right? And I think there has been some work on baboons, and this is something that's special about menopause. There's something about menopause that I don't know. So I think there's a lot of really interesting things to be done there, from everything from the actual empirical, like just description of what's happening through to the evolutionary theory. Fascinating area.
I have a question about this, where should you draw the line, right? And as you were saying, parasite or pathogens, they evolved to be closer to human. So how do you think like zoonotic diseases interact with all this? Because they're gonna not start being close to humans, but they might be adapted to a pig or a bird or whatever. So would males have an advantage, for instance, in when you have a new zoonotic disease emerge?
It's a really great question. So this is one of my favorite research areas. There's a paper that Greenbaum et al. In 2009, which shows that influenza, like if you look at the 1918 influenza, it starts off with a massive CPG ratio. It drove massive cytokine storms. The CPG ratio goes down, 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 presumably because it's adapting to stop killing people so much. I mean, there's also, people have challenged that result, but it seems like it would be a sensible thing for pathogen to do. It would make sense. And that would also open the door to looking at, for example, all of the avian influenza data. Do we expect there worse outcomes in males versus females? How does this play out? The trouble is that people who go work in poultry farms are always men. So it's very hard to probe that data. I mean, there has been some, like Sarah Covey and others have done some lovely work sort of starting to look at this, but I think we don't have a comprehensive enough picture yet.
I can ask one more question. Microphone anyway. I was interested what you said right at the beginning, that around age 7, that seems to be when we're honing in. I was interested, is there any data already then of what happened to those kids that were around the age 7 when the whole world went on lockdown, so they didn't get that exposure to those flu viruses around that age?
Well, we're not enough time.
Right. Are people looking into it though?
I mean, there's so many interesting things. So we work a lot on enteroviruses, and EVD68 is one of the sources of relative of polio, and it occasionally paralyzes children, so it's a terrible thing. And it had pre-pandemic very clear biennial cycle, so an outbreak every other year, which is a very clear signal of immunity, right? Like if you have an outbreak every other year, that means that something's keeping it down. Between years, probably that there is immunity in the population. 2 years of the pandemic, nothing. And you would predict a build up of susceptibles in a vast upsurge. And which we predicted and very happily happened. Well, not very happily, but it did happen. We didn't get it wrong in terms of our prediction. I mean, it was terrible. People were, anyway. But the really interesting thing, so there was more terrible for RSV, actually. It was not so terrible for EVD68, because although the infections came up, the disease did not. So not as many children as anticipated were being paralyzed. And which suggests that there's something pretty intricate going on in terms of either, maybe the best case scenario would be the virus had evolved into something more benign for some reason. Seems a little bit unlikely. It could be something about if it's a second infection that's worse, if there's an age profile piece, I'm not quite sure. So we're doing that alongside a vast cohort study of all sorts of other infections, so that should open the door to also looking into questions we do have that flu antibody titers, and we're using phage display stuff to pick apart what the immune memory is doing. But it's a little early for results on 10-year-olds.
All right, so let's thank Dr. Metcalf.